Good to see everyone and welcome to OsteoBites. My name is Christina Ittoma and I am mom to OsteoAngel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we're talking with Dr. Ben Nacev, and he will share updates from the clinical sequencing of 2,138 bone and soft tissue sarcomas. Thank you so much, Dr. Nacev, for joining us on OsteoBites today. We're thrilled to have you.
My pleasure.
A little bit more about our guest today. Dr. Nacev is a physician scientist with an interest in sarcoma biology, and his work focuses on understanding the causes of sarcomas with the goal of developing new treatments. Dr. Nacev completed his MD and PhD training at the Johns Hopkins University School of Medicine, where he also trained in internal medicine. And he moved to Memorial Sloan Kettering, where he completed his Medical Oncology Fellowship and later joined the faculty before recently moving to the University of Pittsburgh School of Medicine and the UPMC Hillman Cancer Center, where he's an assistant professor of medicine. Welcome, and welcome to everyone joining us today. Please feel free to add your questions for Dr. Nacev to the Q&A, and we will make sure he gets to those. And before we get started, we have a couple of announcements. If you haven't already, please subscribe to our monthly newsletter called Connective Issue. It includes info of interest to our osteosarcoma community, including recent news and papers, open trials, job postings, info about MIB events and programs, upcoming grant opportunities, and also our upcoming OsteoBite schedule. So to make sure you get all this info delivered to your inbox, just go to our website and hit the Join Our Community button to subscribe. And Turbo is our virtual tumor review board for osteosarcoma. Our next meeting is November 9th at 5 Eastern. If you are a clinician or a researcher with an interest in osteosarcoma and would like to be added to the calendar invite, or are interested in submitting a case, just fill out the form on our website and I'll put the link in the chat. And currently we're meeting every other month. So after November, our next meeting will be in January. And if you're in the Houston area, MIB is heading your way. We are going to be there on November 30th for an MIB mingle, 5 to 7 p.m. at the Blossom Hotel right across the street from MD Anderson. And everyone in the osteosarcoma community is invited. And I'll put a reg link in the chat as well. And we'd like to thank the sponsor of this episode, BTG Specialty Pharmaceuticals. BTG provides rescue medicines typically used in emergency rooms and intensive care units to treat patients for whom there are limited treatment options. They're dedicated to delivering quality medicines that make a real difference to patients and their families through their development, manufacture, and commercialization of pharmaceutical products. And their current portfolio of antidotes counteracts certain snake venoms and the toxicity associated with some heart and cancer medications. So thank you, BTG. And Dr. Nacev, I'll turn it over to you now and you can get started and start sharing your slides.
Well, thank you so much for that really kind introduction and the opportunity to present today. The work that I've been doing is really aimed at trying to understand genetic events across a range of bone and soft tissue sarcomas. So I'll describe our experience with that large scale project, but really try to highlight some findings and questions that have come out of this work with respect to osteosarcomas. As you heard, I recently just moved to the University of Pittsburgh, but I want to highlight that the work that I'm showing you today was done back at Memorial Sloan Kettering, my previous institution, with a number of fantastic colleagues, collaborators, and friends there. So as you heard through the last iteration of OsteoBites and likely from other sources as well, we really think of cancer as a genetic disease. Our understanding is that all of the biologic aberrancies and misfirings that have to occur to create a tumor are really seated in events that occur in the DNA of the cells from which the tumor derived that cause the genes encoded by the genome to malfunction and drive the cancer phenotypes. The genetic events in sarcomas are really very highly varied, and the variation and heterogeneity there really mirrors what we see at a pathologic level within the field of sarcomas. So the WHO organizes a panel of expert pathologists that come together every few years, and they help us to classify bone and soft tissue sarcomas into different diagnostic groups. And in the latest iteration of this WHO classification, there were over 170 different subtypes of sarcomas, most of which were actually malignant. So osteosarcomas are one of these 170 different types. Actually, they compose a few subtypes within this group, but really just a subsegment of a very large and varied group of tumors. What we're learning now, and really still in the midst of fleshing out, is that the genetic events that make up this heterogeneity are really as varied as the tumor types themselves. But in an effort to characterize the types of genetic events in sarcomas into manageable groups, historically, we've thought of three main types of genetic alterations. One type is associated with translocations. So what this means simply is that a certain chromosome breaks and is fused back to another piece of a chromosome in a way that two genes that were not next to each other are now juxtaposed, so that in certain circumstances, when these genes are ultimately transcribed into RNA and then translated into proteins, there are now two pieces of proteins that are fused to each other to create a fusion that can drive a novel function. And in the case of cancers, this is a function that causes the tumor cells to grow. So a very canonical or classical example of this is Ewing sarcoma, in which we often see EWS fused to FLY1, for instance. Another main category of genetic events in sarcomas are those that have copy number changes. And what I mean by copy number changes is that either there's a gain or an amplification of a gene, or a deletion or a loss of a gene that affects its function. A classical example of this is dedifferentiated liposarcoma, in which we see amplifications of two genes. Classically, one is called MDM2 and the other one is called CDK4. Still, other classes of sarcomas are thought to be more mutation driven. And usually what this means is that there is a relatively small change within a gene that creates a protein that's either gained or lost activity. Classical example of this is gastrointestinal stromal tumor, in which mutations in a receptor called KIT cause the cells to grow when they shouldn't and generate a tumor. So efforts have really been ongoing to further define these categories and further flesh out all the myriad changes in the DNAs that we see across different sarcoma subtypes. And one really important effort in this regard was the Cancer Genome Atlas dedicated to sarcomas, which was published about 5 years ago. And in the TCGA, the investigators looked at 206 sarcoma samples across 6 different subtypes and really did an in-depth analysis genetically in terms of transcription of genes and even things like DNA methylation to get a very deep understanding of the events within these sarcoma subtypes. And I'll show you just a few examples again of these big buckets of changes from the sarcoma TCGA data to drive home what we just discussed. So returning again to dedifferentiated liposarcoma, these are data from the TCGA. And I've circled here MDM2 and CDK4, which are amplified as depicted by this large red spike here. But even though I told you that MDM2 and CDK4 amplification are really pathognomonic or classical for D-diff liposarcoma, I think you can appreciate that there are many other red spikes here indicating other recurrent amplifications, and there are many blue spikes on the other side indicating deletions that are also observed recurrently in D-diff liposarcoma, really driving home the idea that even though we have some understanding of the events that are occurring, there's a lot more work to be done in terms of identifying and understanding additional genetic changes. Looking at the mutational profile from the TCGA, this is an oncoprint where you can see any box here indicates a mutation, and these are organized by cancer type. I think at this 30,000 foot view, you can appreciate that there are certain types of significantly mutated genes that are recurrent. When we dive into these, we see a lot of alterations in genes like TP53, ATRX, RB, for instance, which is something that the TCGA really highlighted, though was not the first place to demonstrate these recurrent alterations in these particular genes. Another important piece of data that emerged from the TCGA came from comparing sarcoma samples in these colored spots to other cancers, including carcinomas and some hematologic malignancies in these black and white spots. And they compared on the x-axis, the number of amplifications versus the number of mutations. And you can see that the sarcomas really are outliers in terms of the number of amplifications. Those are the gains or losses, I should say, copy number changes compared to other cancers. And you can see they're very low on this axis, this vertical axis, compared to many other cancers in terms of the sheer number of mutations that they have. So that, again, these are the data for those 6 subtypes included in the TCGA. But one of the things that we were really interested in was addressing some outstanding questions that were still existing in the field after really important genomic sequencing efforts like the TCGA had been published. One question for us was, are we able to identify additional drivers in rare subtypes? And by driver, I mean genetic alterations that are likely to be promoting tumor growth. Are we able to identify similarities or differences in biologic pathways between the subtypes? So not just looking at the gene level, but saying certain biologic processes in general, maybe through events in different genes in the same pathway are being altered. And then lastly, can we highlight any new avenues for ultimately pursuing preclinical studies for new therapeutic opportunities? And for us, we really saw a need for a broad single platform comparison across many histotypes or subtypes to delve into the complexity of these 170 different subtypes of sarcomas in the WHO.
Hey, Dr.
Our
Nacev.
goal. Yes,
So
please.
on the slide prior with the quadrants, I didn't see OS on there. Is it?
Yes, it was not included in the TCGA.
Okay.
That's a good point. Yeah, so there were no bone tumors included in the TCGA.
Okay.
But we have data on OS from our study, so I'll show you how we've been able to update that plot as we move forward. So for us, if we could execute such a study, our goals were to be able to generate mechanistic hypotheses from studying these mutations. In other words, by observing changes in tumors, what questions can we then take back to the lab to really try to investigate how these genetic events are changing the biology of sarcomas? And the second goal for us is really to try to improve modeling, to represent the landscape of alterations that are occurring within the tumor setting so that we can better model these in the laboratory. And then lastly, to create a shared resource for the field. We didn't intend for this project to be a comprehensive final publication. We wanted this to be a collection of data, a description of data that we then make available. And I'll show you at the end some steps we've done to do that. So our approach here was to use a targeted sequencing platform. In the latest iteration of this platform, which is called MSK Impact, there were about 500 cancer-associated genes included. Now, this is only a very small subset of all the genes in the human genome, but these have been curated to try to capture genes that are associated through other work with pathogenic changes in cancer. So this panel is notable in that it's able to identify somatic mutations, copy number changes, and rearrangement. So hitting all those 3 main categories that I showed you at the beginning. And I've highlighted the word somatic here because, for those non-clinicians or scientists in the room, this really means that we're looking at changes that are happening just in the tumor, not in changes in the DNA that were passed on through the generations in a family or acquired very early in embryonic development. And the way we were able to ensure this was to make sure that we had matched tumor and normal controls with the idea that the normal tissue controls would capture any changes in the DNA that were passed on through generations, and we're only looking for changes that occur in the tumor. So very briefly, the pipeline for MSK Impact is that in the clinic, I or one of my colleagues would consent patients for participation in this research study. We would then extract DNA from the patient's tumor and then a matched normal control. In almost all cases, this was normal white blood cells, which were isolated from a tube of blood that was just drawn through venipuncture. The DNA was extracted and then underwent sequencing and analysis. And then finally, a report was generated, and the data would go both into the patient chart and then be available for research studies such as this one. The cohort we were able to include in our study was quite broad. So we had 2,100 roughly sarcoma samples that were from unique patients. And these were from 45 different subtypes. Again, by comparison, the TCGA had 206 samples. with 6 or 7 subtypes, depending on how they're classified. Now, this is a very different effort. It's a targeted sequencing panel, so I would say it's not as deep as it's certainly not as deep as a TCGA, but we're able to be more broad because of that and really include more tumor samples and a greater breadth of subtypes. So in this analysis, just to remind you, we're able to highlight mutations, copy number changes, and rearrangements. On the right is the distribution of these 2,100 sarcoma samples. And I just want to highlight that osteosarcoma, which is abbreviated as OS throughout the rest of the presentation, represented 129 of these 2,100 samples, so a relatively large cohort of osteosarcoma samples. I'd like to point out here that the collaboration and efforts from Dr. Christina Antonescu, who is a leading sarcoma pathologist who really helped us to make sure we had as accurate classifications of samples as possible, since really grouping the samples that we had into specific categories of sarcomas was really important for us to generate robust conclusions. I'll also say that one strength of a single institution collection of samples like this was that a small team of pathologists, including Dr. Antonescu and our colleagues, were the ones who were ultimately signing diagnoses to all of these cases. So there was a unified approach to the classification from our expert group. Walking you through a little bit more of the cohort, here I'm showing you in each row different sarcoma subtypes. You can see here the number of samples in each group. We're able to collect outcomes data here, and then also a lot of data on where the sample was coming from in terms of tumor site, whether or not this is from a primary metastatic site. We have a lot of demographic information as well. I'll just highlight here in the orange box the 129 osteosarcoma cases. If you look here, most of the cases here were from primary sites. Vast majority from extremity anatomic locations, as you predict. And you can see that the median age was well under 25 in our cohort. And I'll just point out that we included overall both pediatric and adult cases throughout our entire study. One of the first questions we wanted to ask was, how good is this targeted panel doing at capturing drivers? So that's what we're depicting here. Anything in the dark gray indicates that there's at least one driver event captured on the impact panel. And as you can see, and sorry, I'll come back to that in a minute. And we also looked at the fraction genome altered, which is really a surrogate for how many copy number changes do we have. And we looked at tumor mutation burden as well. I'll come back to this data in more detail as we move forward. Here again, highlighting for you the osteosarcoma cases, and you can see that almost all of them, we had at least one driver that was identified on impact, and for only a few percentage were we not able to identify any oncogenic events that we could predict. And I'll just highlight here and going forward, except for when I specifically point out an exception, any data that I'm showing you is filtered for genetic events that we think are oncogenic. And what I mean by that, for the non-scientists, is that these are predicted to disrupt function of the protein encoded by the gene in a way that we believe potentiates or promotes the growth of the tumor. And this is all from pre-existing knowledge outside of this study that we're able to make those classifications. We also looked at something called cancer cell fraction in order to get a sense for whether or not any of the non-drivers are actually potentially interesting to look at in the laboratory to attribute a function that we don't yet assign. So to explain that in a little bit more detail, each column here is a different sarcoma subtype. The dark colored circles are the drivers. And if we see these dark colored circles up near the top, near 1, that means that almost all the cells in that fraction were harboring this driver mutation, which is what we expect in this setting. And then the light gray are what we call variants of unknown significance, simply meaning that we don't know what these changes are doing, whether they have a function or not. And in some cases where we see very high cancer cell fraction of these VUSs, that indicates to us that maybe they actually are functional if all the cancer cells are selecting for them, and they could be candidates for further study in the lab. When we look at osteosarcoma, we actually see a little bit of an exception to the rule where the drivers are quite high. and the VUSs are a little bit lower than in some of the other cancer types that you see here. Nonetheless, I think that a number of these VUSs may be very interesting for study in the lab, and I'll highlight some examples in a few slides. Sometimes I get asked, what's the frequency of mutations per subtype? And this is some data across the board where we've tried to answer that in this study. We've used a few different computational tools to identify subtype specific significant mutations. So what we're doing here is grouping by different mutations, different genes. So for instance, here's TP53, here's RB, here's ATRX, which may sound familiar from that TCGA study. And we've shown that within a single panel, so comparing apples to apples, what's the frequency of oncogenic alterations, for instance, in TP53 in these different sarcoma subtypes. I can show you again, here's osteosarcoma. So 26% of the osteosarcoma cases we saw had an oncogenic alteration in TP53. The rate for RB was about 7%, and you can trace this in ATRX as well. For some of the other genes here, we didn't see highly prevalent or significant recurrent mutations. This is just mutations in osteosarcomas. But to just highlight the power of doing this type of analysis, we were actually able to identify a recurrent mutation in a gene called FLT4 in another type of sarcoma called angiosarcoma. And one of the important things here is that this particular set of mutations were classified as those variants of unknown significance. In other words, people weren't really able to say with certainty that these alterations were potentially contributing to the behavior of angiosarcoma. But when we really dived in to look at where these mutations occur on what's called a lollipop plot here, you can see that the longer stalks representing more mutations with the orange stalks, specifically in angiosarcoma, are piling up in certain locations in the FLT4 protein. You see them in the sort of the active part of this transmembrane receptor, which is called the kinase domain. You see them in a very short stretch of the C-terminal domain of FLT4. And when we see a prevalence of mutations occurring in a certain region of a protein as compared to a smattering of mutations in other sarcoma types, which are represented by these gray stalks, this really indicates to us that there really may very well be an important function of these events in this gene of angiosarcoma. Again, driving home that this technique is able to highlight new hypotheses that we can test in the lab.
Hey, Dr. Nacev, we had a question come in. Would you expect that all cells in the tumor would have the same genome signature, or are there a collection of clones with different genome signatures? And if they're different clones, how does that impact your analysis?
That's a really great question. So without a doubt, there's significant heterogeneity within the tumor, and different clones can grow out within a single tumor. So I think that's one thing that's really an important point as we think about the analysis. What I'm not reporting here or built into the algorithm is sort of taking into account this allele fraction is one way to think, one surrogate for the tumor purity. And we also include tumor purity within our data set, which is really more of a marker for not the heterogeneity but within the tumor, but in a tissue sample, how much is tumor, how much isn't, which is another factor when we have to do this. So it's all taken into account in the analysis, but absolutely there are likely different clones within the tumor, and we can't separate those out necessarily in this analysis. Any other questions?
Yeah, actually, there have been a couple popping up.
Great.
One related to the VUSs. So I believe most mutations are VUSs. How will you utilize those to identify new important functions for those genes?
Yes, this is a great question. I think by diving deep into the VUSs, like that FLT4 example that I just showed you, to ask, are they scattered throughout the gene? Are they in certain hotspots? May highlight candidates that we then really need to model in the laboratory. And by that, I mean, generate cell lines where we reproduce the mutations, understand how they affect the function of the biologic pathway in which they're involved, how they affect tumor growth, and then ideally, go forward to create animal models to further study these changes. Of course, it's not always possible in all subtypes, but aspirationally, that would be what the path one would take.
Great. And one more question before you move on.
No, great. Yeah.
Is your pipeline able to identify rearrangements that inactivate genes like intron 1 rearrangements in P53?
Yeah, so if a gene is on the panel, we should, in most cases, be able to identify rearrangements. It's not 100%, but it certainly gives us the capacity to do that. I actually haven't included much data on the rearrangements in this talk, but at the end, I'll show you how you can access the data set and look for rearrangements through the CBio portal. And there's additional information in the manuscript that accompanies this.
Great, thank you.
Great. Any other questions?
Not for now.
Okay. All right. So another analysis that we were able to do with the mutational data in hand was really to turn to this question of tumor mutation burden and microsatellite instability. So the reason why we look at these two features is that in other cancers, in certain carcinomas, for instance, high tumor mutation burden or microsatellite instability are predictive biomarkers for response to checkpoint inhibitors. Now, we've known going into this from the TCGA and other studies that in general, the mutational rates in sarcomas are low. But we're able to really look at that across many different subtypes and get a sense for the distribution of mutation rates. So TMB is simply number of mutations over the amount of genome sequenced. Since the genome was the same size and all of us humans, the denominator is the same. So really the TMB is just a surrogate for the number of mutations in general that we're seeing in these samples. And so what we've shown here on the left is the TMB for each of the sarcoma subtypes that are listed along the bottom. And I think you can appreciate that the means, as indicated by these solid bars, are relatively low, far less than the TMB of 10 that tends to be the threshold for thinking about predictive biomarker in carcinomas. But as you can appreciate, there is a long tail for a number of these subtypes, including including in osteosarcoma, which I've indicated with the green arrow. So again, the most of the samples have a TMB less than 1, but there is this outlier cohort that really tails upwards. And we really don't know within this setting of sarcomas whether the threshold of 10 is the right threshold to be thinking about a predictive biomarker, it may be lower. So simply providing this information we thought would be interesting for the field. And you can see that tailing effect in a number of other subtypes as well. On the right, we looked at microsatellite instability using this computational MSI sensor score as a surrogate, but 10 is the threshold number here as well. And you can see out of the entire cohort, there were only really a handful of cases that were MSI high, and only 4 that were actually confirmed with an orthogonal assay. So microsatellite instability is vanishingly rare in bone and soft tissue tumors, but long tail of TMB is a little bit more common. I'll show this briefly. We also had the ability to look at mutational signatures to understand where these mutations are coming from in tumors. We used a technique that really required a relatively high number of mutations to derive the signatures. So most of the samples that we had, we couldn't analyze in a robust way, which is why you see numbers here are quite small. So for instance, there were only 9 osteosarcomas that we could analyze for mutational signature, meaning that 120 of the cases we couldn't. That said, for some tumor types like angiosarcoma, we see this preponderance of red, which correlates to a UV signature, which supports some previously known, previously reported data. In UPS, we see a little bit of a red signature as well, and a blue aging signature. Though again, for the small number of osteosarcoma cases, there's really not a standout pattern that we can observe. I want to shift now to talk about copy number changes after having gone through mutations. This is a very busy 30,000 foot view of how we are able to look at copy number changes in this cohort. So what we have is the chromosomes displayed along the x-axis. Anything that's blue is a loss of a segment of the chromosome. Anything that's red is a gain of a segment of that chromosome. So I think you can appreciate some examples like GIST, where there's periodic blue patches, which indicates scattered loss of chromosome arms. Whereas in osteosarcoma, you can see that there's a fair amount of red and some intermixed blue, which really supports the idea that osteosarcoma has many copy number changes. And speaks to what we already know, which is there is a very sort of reorganized or rearranged genome within osteosarcomas. Now, I certainly can't make too much sense out of this very high-level analysis. So what we tried to do was to apply some statistical and informatics analyses to ask the question of, are there certain segments of certain chromosomes that are recurrently altered in a statistically significant way? And that's what this panel attempts to do on the left. So anything with an asterisk in it indicates statistical significance. And we're looking on the left at chromosomes. And on the right, we even tried to narrow this down to individual genes. So again, highlighting osteosarcomas along the bottom, we can see, for instance, there's a significant gain of one arm of chromosome 5 and chromosome 8, and losses of chromosome 10 as well. And when we look at individual genes, most of what we can pick up are in MYC, which we've confirms data from other studies, as well as a few genes where we don't really know the function. And I'll come back to one of these in core 1 down the road. And we also see loss in a few other areas as well, including TP53 and RB. But hopefully this gives you a little bit of a flavor for how we can delve into the copy number data. An additional analysis that we could do, and I think we've probably applied this for the first time in osteosarcoma, but I don't know that for sure, is to look at whole genome doubling in these populations. And the reason why we wanted to look at this is that a few years ago, there was a pan-cancer study that looked at whole genome doubling through copy number changes and other events that demonstrated that patients who had whole genome doubling in their tumors unfortunately did less well than patients who did not have whole genome doubling. And the field is really still trying to understand the reasons for this, but the authors of this initial paper described that the percent of whole genome doubling correlates with higher proliferative index. So maybe by having more copies of genes that are important for cell growth, this can lead to an increased growth rate that can drive the worst outcomes in this population. Having read this and observed this in other populations, we wanted to understand where sarcomas fit within this whole genome doubling landscape described earlier. So what you see here at the bottom is our data intermixed with the data from that prior publication I was showing at the top. So the light gray are the cancers in the prior publication, and the dark gray or kind of greenish colored are the sarcomas from our study. So as you can see here, there's really a bimodal distribution. There's a group of sarcomas that really have very low rates of whole genome doubling compared to other cancers. Included here are, Ewing sarcoma, synovial sarcoma, for instance. But at the other end, there's another collection that has very high rates of whole genome doubling compared to other cancers. And this includes, at the second from the left, osteosarcoma. So a very high rate of whole genome doubling, at least half the samples in our cohort, which really is at the high range of all cancers. We want to know within our population of sarcomas, does whole genome doubling actually predict outcomes like it did in the pan-cancer analysis? It's a little bit of a complicated plot, but what we did notice was that for samples that were sequenced from metastatic sites, that's the orange shades, if there was whole genome doubling, that's the darker orange. Generally, unfortunately, the survival from the time of sequencing was less than those cases where their whole genome doubling was not observed. We tried to break this out on individual subtypes. We did the only subtype where we actually saw a similar correlation once we broke down the data down into smaller numbers was an undifferentiated pleomorphic sarcoma. Again, looking at the orange, this trend of worse outcomes in the case of whole genome doubling was significant compared to those cases that did not, within the metastatic group. Just to pivot a little bit now to think at a more higher level of how we're organizing these events in ways where we can work towards addressing mechanistic hypotheses in the lab or building better models that I alluded to, was that we wanted to look at each of these genes not as individual entities, but really a collection of pathways of biologic function. So that's what we've done here. So each row is a different sarcoma subtype. Here's osteosarcoma here. And each column is a different biologic pathway. The darker the purple means the higher the rate of oncogenic alterations within that cohort of samples. So on the left, oops, sorry, I'm just highlighting the osteosarcoma here. On the left, these columns are cell cycle and TP53. So you can see across the board, a relatively high rate of alterations, which we would expect. On the rightmost column, this is a collection of genes involved in epigenetic regulation, which I'll say more about in a few minutes. And as you can see here, there's actually a relatively high rate of dark purple here, indicating that the epigenetic pathways are recurrently altered in osteosarcomas and other sarcomas within our cohort. So we use this panel view to allow us to break down these pathways in more granularity, and I'll show you some vignettes of what we've learned. For instance, we identified TERT alterations, and this is involved in allowing cells to continue to maintain telomeres, which are important for allowing continued cell division. And we identified for the first time that about 40% of a very rare type of sarcoma called intimal sarcoma have amplifications of the TERT gene, which may be important for their biology, to give one example of how we can break out these pathways. And to show you one more example, we're able to start with a general pathway, the PI3 kinase pathway, which represented one of these columns at the bottom, and say, let's break this out into individual genes and look for similarities or differences between different sarcoma types that have alterations in this pathway. So along the top, each of these columns is a different sarcoma subtype. Osteosarcoma isn't included here in this example, but I wanted to show it to you so you can get a sense for the type of analysis we can do. And these are all the genes that make up the pathway. When we look at myxoid round cell liposarcoma, for instance, we can see that there's loss of this PTEN tumor suppressor. And as you can see, looking across the row at these other sarcomas, most of them have similar rates, or a few of them have similar rates of loss. But when we look at PI3 kinase, or PIK3CA, which is an oncogene, we can see that myxoid round cell liposarcoma really stands out from the rest of its neighbors in terms of having activating mutations here. So we're able to highlight, again, these similarities and differences once we break down these pathways, which I think is important for modeling or even thinking about therapeutic implications. So what I'd like to do over the next few slides is really focus you back in on this epigenetic pathway, on these epigenetic pathway alterations within osteosarcomas and some other sarcoma subtypes. As a way to introduce that, I just want to say a few words for the general audience about what epigenetic regulation is. So it's something that's critically important for normal physiology, and we've also learned that alterations in these regulatory pathways can be very important for driving cancer biology. So on the left is a figure from Conrad Waddington, who described something that we've now used as a metaphor for the epigenetic landscape many years ago. He envisioned this little P at the top of a slope, which represented an uncommitted progenitor cell, which rolled down this slope and encountered these different forks and channels that helped dictate its behavior. Did it turn into one cell type or a different cell type? But what we've also appreciated is that the epigenetic landscape can be disturbed such that cells can either differentiate into their normal tissues or behave aberrantly, become cancer cells. So over the many decades since Waddington, we've learned a lot more about the molecular features of what epigenetics, what this landscape is, and what are the factors that regulate epigenetic landscapes. To break this down from one point of view, we think about the genome as not being sort of a string of DNA, but instead it's wrapped around nucleosomes, which are composed of proteins called histones, and that these nucleosomes are substrates for a lot of regulation by a number of different classes of regulators, including a class of proteins we call writers, which deposit post-translational modifications on nucleosomes, erasers that can remove these, and reader domain proteins that can really bind to these PTMs in a chemistry and site-specific way to bring in other machinery to the genes in this region, like those that activate transcriptional programs or repair DNA. So a lot of different signals like environmental stimuli, energy and metabolic states, and developmental signals can impinge upon all these writers, erasers, and readers to dictate their function in controlling cell cycles, so cell division, responses to DNA damage, or transcriptional programs, which help determine cell fate in a way that Waddington envisioned many years ago. So there's some classical examples for how genetic events and epigenetic regulators are important for sarcoma biology. I'll just give you one example here, which is epithelioid sarcoma. It's known that a certain class of remodeler, which can shift shift the position of these nucleosomes to mask or reveal different DNA sequence, is recurrently altered. In this case, SMARCB1 is a gene that's lost in epithelioid sarcoma, and you can tell that because there's no brown staining in these cases, except for in some normal tissues. And by understanding the biology of SMARCB1, we know that the complex which normally harbors SMARCB1, which is called switch NIF, and again is a remodeler, when this function is lost because of SMARCB1 deletion in epithelioid sarcoma, this creates a dependency in a PRC2, which is one of these writer complexes, such that if we develop EZH2 inhibitors, these can preferentially stop the growth of cell lines that harbor SMARCB1 alterations. And in fact, this EZH2 inhibitor was developed clinically as tazemetostat, and here's some data from a phase 2 clinical trial, which showed about a 15% response rate in patients who had epithelioid sarcoma and received tazemetostat. And the FDA actually approved this drug on the basis of this trial. So I show this example really to drive home the point that if we can understand genetic alterations in epigenetic regulators, or first identify them, then try to understand them biologically, there's potential to develop new therapeutics for sarcoma patients. So with this in mind, we really delved further into our data set to look for emerging examples, additional examples of genetic events in these epigenetic regulators. So that's what I'm showing you down here at the bottom. Each column this time is a different sarcoma subtype. Again, the dark purple means that this is predicted to be an oncogenic event, and where the darker the purple indicates the more often that's happening in a certain sarcoma subtype. And I've broken down in the rows loosely on the basis of these different classes that I showed you up here at the top. One vignette here about something that I think we were able to show for the first time in our analysis was looking at a very rare sarcoma subtype called uterine adenosarcoma. We only had 14 cases in our cohort, but as you can see, more than half of these had alterations in chromatin remodelers, and about a third had alterations in histone modifying genes, again, which was not appreciated before this effort. And this is the same switch NIF complex that was affected in epithelioid sarcomas, the example that I just showed you, that we don't know the function of these events yet. We're also, of course, interested to see what the changes were in osteosarcoma, again, highlighting here that we see a relatively high rate across different categories. And when we break this down, a few interesting things emerge. So I know this got very busy, and what I'll tell you here is there's no oncogenic filter here. So instead of just focusing on changes that we know to be important or potentially important from other cancers, I sort of put on my hat as a discovery-based scientist and said, I just want to know what the changes are. Let's think about which ones we might want to take to the laboratory and model in ways where we might be able to inform the entire community about the activity of these changes. So again, focusing on osteosarcoma, in the orange box, I think you can appreciate that there's a relatively high rate within these large categories. And when we dive down a little bit deeper, we can see this particular gene called NCOR1 is altered in 21% of this large cohort of samples. So what does NCOR1 do, and what are these alterations like? Well, almost all these alterations are amplifications. So there's a gain here of the NCOR1 gene. This indicates that likely there's more protein made, which really has to happen in order for this to have a functional effect. At the bottom here, I'm showing you data on NCOR1 expression with amplification from the TCGA. Now, I told you at the beginning that the TCGA doesn't include osteosarcomas, but it's really a surrogate to ask, when NCOR1 is amplified in a sarcoma, do we see more gene expression? And we do. Okay, so this suggests that this may be equally overexpressed in osteosarcomas. What do we know about NCOR1 already? Well, it's a transcriptional co-repressor, and it complexes with an HDAC. So this is a member of one of those eraser complexes that can change the chromatin landscape. It's been known to regulate mesenchymal lineage transcription factors in other studies. And in non-sarcomas, it can suppress differentiation when it's overexpressed. So all of these features may have implications for vital chromatin functions in osteosarcomas where this is amplified. But truth be told, we really don't know the function of NCOR1 in osteosarcoma, and needs to be studied in the laboratory. But I think there's some potentially intriguing hypotheses that have come out of this sequencing effort with regard to this gene. To pivot away from epigenetic. Yes,
Sorry,
please.
I had a question about the different buckets that you mentioned for the genetic events. So for OS in particular, it looked like there was heavier concentration in the histone modifying and the chromatin remodeling. And does that
Yes.
have any implications? I'm just curious if there are particular characteristics of those different types of genetic events.
Yeah, so it's a good question. So I think it's hard. I think probably, the implication I think you're probably asking about are on sort of the cell level or the tumor level, right? In terms of the behavior or sensitivity to different therapies, that we certainly don't know yet. And the first step to me would be to understand how these alterations affected these different functions, right? So how is chromatin remodeling affected within cells? How are the histone modifying enzymes affected in their activity within those cells? And then knowing what we know about chromatin's role in controlling things like, again, DNA damage repair, differentiation, what does that say at the cellular level? And then once we build the cellular models, how can we look for vulnerabilities where we might be able to think about exploring drugs preclinically to then move ultimately back to the clinic. So this is all very much hypothesis generating. I'm conscious of the time here. I want to be able to show you at the end how you can actually dig through this data for yourself. So I'm going to zip through these last two slides and I'll just show you this summary. So again, we reported about 2,100 sarcoma samples over 45 different subtypes, and 129 osteosarcoma samples were included here. And again, just to highlight a few things from our findings, I've shown you that NCOR1 amplification is very common in osteosarcomas, that there are high rates of whole genome doubling in sarcomas, including in osteosarcoma. We don't yet know how that may affect the biology of osteosarcomas. And then the last one I didn't get to, but happy to chat about genomic clusters in the Q&A if people are interested in. But what I really want to highlight too is that our goal in doing this study was not just to analyze the data for the purposes of reporting it in one paper, but to create resources for the community. And really that means everybody. So the manuscript is published, so there's some more details there if you want to dig into this. But we've also made the data available on CO Bioportal. You can use the QR code to link to it if you want to take a picture or a snapshot of it now. But what I'm going to do just for a moment is walk you through the data or how you can look at the data. I'm just going to have to switch my screen sharing, I think, for you to be able to see this. Bear with me for 1 second so I can share the correct screen. Okay, can you see a CBAR Bioportal page? Okay.
Yes.
Okay, great. So this is the link that the QR code will take you to. Bioportal was a tool created years ago by a team at Memorial Sloan Kettering. It's completely open to the public. This is just a page for our study, but many other studies are here. So you can see this is it's kind of a dashboard for all 2,100 samples from our study. The key thing, or one thing for you to recognize, is these great tutorials here. So I'll show you just a few pieces of functionality really to familiarize you with what's here. But you can click on the tutorials that'll teach you how to go through this yourself. So this is a dashboard, like I said, for a study. You can see in this box all the different cancer types, sarcoma subtypes that were represented, and it's rank ordered. Again, you can see osteosarcomas here, which is relatively at the top of the list in terms of prevalence. And all these different boxes show
mutated genes, structural variants. So those are the rearrangements, copy number changes. And if you want to just focus on one cancer type, you just have to click the box. So now we're filtering only for osteosarcomas. As you can see, for mutations, TP53 is the most altered, and we observe it in 25% of cases. RB is next, and you can go down the list. Again, this is not filtered for oncogenic, so that's something to keep in mind. You can set up that filtering and learn how to do that through the tutorials. Same thing for copy number changes. NCOR1 is actually at the very top of the list. Again, about 20% of these cases had amplifications, and that's the most change out of any gene that was included. You can also see other data here like TMB that we talked about. You can see different categories in terms of mutation count, and I'll allow you to look through this. But what I want to show you is how you can actually put in a particular gene that you're interested in. Let's say, for example, MYC, you can type it into the search box and query, and you can get all sorts of additional information. Again, this is pulling entirely from the study and the genomic sequencing that we undertook. So you can see across the board, there's about a 10% rate of MYC changes in our study. And you can tell that these are all amplifications by the key at the bottom. If you say wanted to look at the individual sample level, you just hover over and click the sample, and it pulls up a page for this particular sample. Of course, everything here is de-identified. And you can see other mutations that occurred also in this sample, along with the MYC amplification. You can see other amplifications as well. And there's a lot of data embedded in here. Can you guys still see me going through all these different windows, or did I lose you when I switched?
No, we can see it all.
Okay, hold on. I just need to move this panel for a second so I can get back to where I was. All right. I know we're about time to start the Q&A, so I'll just show you a few other things that you can look at here. You can look at different pathways. You can see that whether other genes in the same pathway as MYC are altered. In this case, there's not. MYC sort of stands on its own. And you can look at other things like outcomes data, et cetera. But again, I really just wanted to let you know that these data are on the CBR portal, and it's a really wonderful tool to dive into to get to know the data set. So I think with that, let me just switch back to my last slide so I can thank the correct people. Of course, there are a lot of people to acknowledge with this study. And as I highlighted, this was done in collaboration with a very large team back at Memorial. Shalia and Francisco were really key in the bioinformatics analysis, and Sam Singer, Bill Tapp, and Christina Antonescu were instrumental from a leadership standpoint. And we had many collaborators across different departments. But I really want to acknowledge all of our sarcoma patients and families for participating in this study so that we can learn more about this disease and hopefully do a better job in studying it in the lab and bring treatments back to the clinic ultimately. So with that, I'll stop sharing and just thank you all for your attention and look forward to discussing.
That was fantastic. Such rich information, and especially also wonderful that it's in the CBio portal so people can really do deep dive. Another question from our audience, are higher rates of whole genome duplication associated with widespread copy number structural variations across sarcomas?
So is whole genome doubling associated with widespread copy number changes? Yeah, so I think the copy number changes are to my understanding, what's driving the whole genome doubling classifier. But we don't know, I think if you're asking, and if we look at a particular case where we said, okay, we observe whole genome doubling, can we track that back to specific genes? That's a good question. We haven't done that. I'm not sure if it's technically possible, but I think that's a very intriguing point.
Dr. Nacev, I'm super excited when you're starting to show, to be able to quantify the mutation types, because we've always heard TP53, MYC, RB1, HRX, and so to start to quantify those, right? Because then that helps us just understand and focus, and then also for risk stratification, because we keep again hearing, but it's just helpful to know the frequency at which these occur. And so you have a pretty good sample size, but there have been some other studies published recently, like around the same time, like Dr. Gounder, also at Memorial Sloan, who looked at the Foundation Medicine data set, which was quite a large data set. I think he had like 460 OS samples in that data set. So I know it's kind of apples and oranges because they're different panels, but I'm just wondering, and there have been some other studies, like I think Dr. Church also published a paper where there were, maybe like, I think over 60 samples, OS samples in that. So to what extent can we start to kind of combine these? Because the numbers start to get a little bit bigger once we can combine to really quantify the mutations.
Yeah, that's a great point. I think, the greater the number of cases, the more accurate we are in really classifying the true frequency. And, even though we had 129 cases, of course, there's certain, limitations and biases within a single institution that, may push us towards a slightly higher or lower number. So I agree with you. It's important to combine the data. I think, unfortunately, the way it often works in science and medicine is that, these types of studies are generally siloed and hard to pool all the data together. I think the best step to doing that is really to make sure everything is public. And that's part of the reason why we put our data up on CBio Portal. Now, you'll notice that on CBio Portal, there are many studies that were not from Memorial Sloan Kettering that are there, the TCGA studies and other institutional studies. So there's nothing that prevents these other efforts from being combined into the CBio Portal so that people can look side by side at all the samples together. So that would be one suggestion, to ask everybody in the community to start to deposit their data in databases like CBio Portal. I think it may be a little bit easier than simply just making uploading the data there. There could be teams I think that may be able to help with incorporating the data into the larger platform. There's also ACR Genie, which can be a place for depositing data. But I think at this point in time, it's really often up to the individual investigators for how they choose to make their data publicly available. But it has to be an ongoing conversation, because I agree with you. The more we can aggregate, the better we'll be at really understanding the true frequency of these events.
And I don't know, I can't remember now if your slides got to this. I got a little preview, so these might have been on the slides that we didn't completely get to, but there are these groupings or clusters of mutation types, right? So you might have TP53 plus RB1, or you might have 3 combination, throw HRX in there. And so how differently do those groupings act? And does it make sense to kind of think of them as groupings versus individual mutations? So I guess what I'm getting at is just knowing 30% have a TP53 mutation. Is that as helpful as knowing that 30% have actually this combination? And does that act differently and behave differently?
Yeah, great question. So I that was one of the few slides I had to skip over, but just to describe for everybody else, one thing that we tried to do was to, in essence, throw out that WHO book and said, we're going to blind ourselves to the histologic subtype, right? And say, let's look at their genetic subtypes, and then cluster this entire population based on their genetic subtypes, and then go backwards and say, well, are all these clusters depicting a particular histologic subtype as we define it now or not? And in some cases, like in well-def-def liposarcoma, that's the case. But in many other cases, in particular osteosarcoma and a few others, there is a very high rate of heterogeneity, where individual samples clustered based on genetics, which tells us that we have to take that into account when we look at outcomes data, when we design studies, when we model osteosarcoma in the lab. Because just as you said, there are some clusters that have just TP53, some clusters that have TP53, RB, and ATRX. And we're able to look at sort of the outcomes data from our cohort, but the more we create subgroups, the less good the numbers are. The smaller the numbers are and the less reliable the data is. So I think that's a limitation, but it's theoretically possible to ask those questions. But for me, I think recognizing those different groupings based on genetics is really important for 2 things. One, to think about how we model these in the lab, right? If you want to study an osteosarcoma line, you need to at least ask yourself, well, which of these subgroups are we studying, for one? And then 2, when we're doing clinical trials, it might be also good to know, can we break down the genetics of the patient's tumors who participate in the trial into these different subcategories? And we really build that knowledge base that you're alluding to and asking about, in I think a very important way.
Okay, great. I'm sure we could spend a lot more time talking about this, but we are up against the time. I did just want to throw out before a quick close, because there was a question from the audience about CRISPR and applications for OS, which I know is not a 30-second answer, but I just wanted to make sure we address that question.
Yeah, I don't know whether that was sort of as a therapeutic strategy or a research tool. For a research tool, it's something we routinely deploy in the laboratory to model these different alterations. For a therapeutic tool, there's a long way to go to get there safely. So that's a short answer.
That was a very good attempt at a quick answer.
Yeah. Thank you.
All right. Well, thank you so much, Dr. Nacev, for joining us on OsteoBites today. We'll make sure we actually include a link to your paper and also to the CBio portal for everyone who's registered. So thanks for joining us and for making it better for sarcoma patients everywhere. More information on this and all OsteoBites can be found on YouTube, on our MIB Agents channel, on our website, mibagents.org. And you can also listen at your favorite podcast place. And join us next Thursday, November 3rd. We're going to be talking to Dr. Dan Regan, an assistant professor at the Flint Animal Cancer Center at CSU, Colorado State University. And he's one of our Outsmarting Osteosarcoma grant recipients from 2022. He'll be discussing his Outsmarting Osteosarcoma funded work on focal adhesion kinase inhibition to improve losartan, sunitinib, immunotherapy, and metastatic osteosarcoma. That was a lot of syllables in one sentence. The registration link is in the chat, and you can find our OsteoBites lineup for the rest of 2022 and even into early 2023 on our website. And if you have any ideas for future topics that you'd like to hear about, please share them with us at events@mibagents.org. Thank you again, Dr. Nacev, for spending an hour with us today, and to our sponsor, BTG Specialty Pharmaceuticals. And thank you all for joining us on OsteoBites. We hope to see you next week when we talk to Dr. Dan Regan. Thanks so much, everybody. Have a good rest of the week.